Uh, Good morning, everyone. Welcome for the second session of day 2 of ISTE AICTE sponsored 6 days online refresher program on leveraging academic research on developing diagnostic kits, vaccines, and drug products for improved therapy management against deadly viruses. Lesson learned from COVID-19. I would like to take this opportunity to express our hearty welcome on our institute behalf to our guest speaker for this session, respected Dr. Kamal Dua, sir. Welcome, sir. Dr. Kamal Dua is an award-winning pharmaceutics and drug delivery researcher who has deep roots in teaching, research, and industry spanning 3 nations and 2 continents. It's my immense pleasure to introduce speaker for this session, Dr. Kamal Dua, sir. Dr. Kamal holds dual doctoral degrees in pharmaceutical sciences and immunology and microbiology, and has completed 4 professional diplomas in the areas of clinical research, product development, drug regulation, and documentation and intellectual property rights. In his role as a senior lecturer in the Graduate School of Health at UTS, Kamal leads a suite of good manufacturing practice courses, where as a program coordinator, his responsibilities include coordinating the academic and research activities for those courses. Drawing on his experience with various pharmacy schools in Australia and overseas, he has blended different modes of innovative teaching with traditional teaching modes to create a flexible, student-centered, and engaging approach to pedagogy. Kamal is also an adjunct assistant professor, faculty of pharmacy and pharmaceutical sciences, University of Alberta, Canada, core member, Australian Research Centre in Complementary and Integrative Medicine, UTS, adjunct fellow, NICM Health Research Institute, Western Sydney University, Australia, adjunct faculty, Manipal College of Pharmaceutical Sciences, Manipal, Academy of Higher Education, India, and research associate at the Australia-China Relation Institutes. He also held a conjoint lecturer position with the School of Biomedical Sciences and Pharmacy, the University of Newcastle, Australia. Kamal's collaboration include working with Dr. Michael John Rathbone, former professor and Dean School of Pharmacy at the International Medical University in Malaysia, and founder of ULTI Pharmaceuticals, a veterinary pharmaceutical company based in New Zealand. Kamal is associated as a consultant on quality systems management and GMP for ULTI Pharmaceuticals. His academic research has contributed significantly to the field of managing chronic inflammatory diseases, especially airway diseases, using novel nanocarriers to deliver potential therapeutic molecules, including phytoconstituents. He has more than 488 publications in reputed journals, citations of 6,656, H-index 31, and I-10 index of 161. Dr. Dua is an active member of many national professional control release society and international Malaysian Society, Pharmacology, Physiology, Pharmacy Council of India, etc. May I now request Dr. Kamal sir for his presentation on investigating interactions of influenza virus infections in asthma and COPD. Please, sir.
Thank you, Popat. Can you confirm that you can see my PowerPoint slide clearly?
Yes, sir. Yes, it's visible.
Okay, great. Thank you so much. Thank you so much, Professor D'Souza, and thank you, Popat, for your nice introduction and for inviting me to talk on this platform in this session. So today I'm going to talk very different actually. So as my lab works in both the areas of the drug delivery as well as on the biological side. So what I'm going to present today is all the biology, what we do here in UTS and the other institutes I'm being associated with. Because currently it's a COVID time and we still being, sometimes we think it has surpassed, it has not surpassed. Every day we have been encountering a new strain of a virus. Virus will never stop, never. Do you think it will stop? No. Virus is a thing which keep mutating. The strain would be keep changing. It is just the more of the discoveries which actually help us. And one of the same thing is with the influenza virus. It was the same time ages ago, what we are seeing currently with COVID in 1918, actually, when people actually affected with the influenza virus. So my talk today is going to be solely focused on flu, what we also call influenza virus, or we also call IAV. In this research, I'm going to talk about why the patients, when they have asthma and COPD, which is the chronic obstructive pulmonary disease, and when they get flu on top of that, their condition really exacerbates. And it's really for those patients, the conditions become to that level, it's hard to breathe and the mortality rate increases. So what else we can do to prevent those kind of the interactions? This is what I'm trying to show in my today's presentation. So before I start, I would just like to introduce myself a bit. So I am from this second largest state of Australia called New South Wales and based in Sydney. And everyone might be aware of the two important hallmark features or the hallmark, buildings or the icons of Sydney called Sydney Opera House and the Sydney Harbour Bridge. They are not very far from the school where I am based at. They are hardly a 15 to 20 minutes walk to this institute, from this institute called the Graduate School of Health, or where the discipline of pharmacy is based. This is an old building. We have just moved to a new building. This is a previous institute called Hunter Medical Research Institute. This is a part of the John Hunter Hospital and the University of Newcastle, where this research, which I'm going to talk today, is being conducted, being associated with that. And I picked that topic because it is very much correlated with the theme of this session, which is all going on for the couple of days. So before I jumped into my data or the research, what we're doing, I really want to show this paper, which is published in Lancet, and that's very clearly showing the great burden of the chronic respiratory diseases in a country like India. So when we really look at this image, you can see that the red color spots are clearly showing that the prevalence is more than 4,000 or 5,000, close to 5,000 per 100,000 people of the respiratory diseases. It is highly prevalent more in the northern part of the India as compared to the central and the south part of India. And that clearly shows with the enormous news coming every day that the number of the medications, what people need every day is increasing. If we talk about New Delhi, the things in winters, what it comes called smog, and it's a combination of the smoke and the fog, where the particulate sits in the fog, and when people breathe, they get the particulates being inhaled, and that can lead to inflammation in the lungs and lead to the various diseases such as asthma, and also maybe down the track to the COPD or even to the cancerous stages. Now, I'm coming to the main core part of my presentation, which is flu. With the basic introduction in 1918, almost 1/3 of the world population, which is equivalent to around 500 million people, were affected by influenza virus. Exactly the same scenario in 1918. What's exactly happening in the last 2 years with the COVID-19? Their estimated toll that at that time was around 50 to 100 million people. And every year, whenever these seasonal pandemics come, they're just being trash the people, and the deaths can be up to 250 to 500,000 deaths worldwide. In Australia, if we look, despite it's a small country, it has almost more than 18,000 of the hospital admissions and more 1,000 deaths per year. If we look at particularly in India, despite this figure is really an old figure, the number has drastically increased now, equivalent to 43 million episodes of the airway respiratory infections, out of which around 4 to 12% is because of flu. But there are so many problems with whatever the current treatments we have for the flu or influenza infection. The first important thing is the influenza virus actually mutates very rapidly, and it changes its virulence year to year. And when it's changed its virulence year to year, it gets really difficult to have a new vaccine every year. So we need a universal vaccine which can manage and mitigate all the different strains. The current vaccines are not effective, and they are only effective when given in the first 48 hours of the infection. And what we normally do when the flu happens, the people normally try to take their own herbal remedies in the first 48 hours, thinking it's just a normal flu. And they try to have, the basil leaves tea or whatnot. And by the time they are presented to the clinic, the condition is at the point when those vaccines will never work. So it are only effective in the first 48 hours. And when we give the combination of the antiviral therapies, this is what we do now, and they are not effective either. When we talk about a particular disease called asthma, according to the WHO, there are 300 million people worldwide, those who are affected with asthma. 2 million of Australian, where about 1 of 10 in adults and 1 in 9 or 10 of children. In Australia, the rate is being increasing every year, despite the figures are old. And in India, it's almost around 18 million asthmatic patient, which is costing around 2.7 billion Australian dollars. What exactly happened in asthma? It is a chronic inflammatory disease, which everyone knows, and the basic biology is very clear that it causes the inflammation in the airways, and that leads to the narrow down of the airways. And because of that, what will happen is when it narrowed down, there's a lot of production of the mucus from the epithelial cells, and that can clog the airway. And that's why when it becomes so narrow, if you compare with the healthy airway, it gets so hard for the patient to breathe. And why it happened? This is all happened because of a factor called airway hyperresponsiveness. And that is because there's a lot of inflammation there. There's a lot of dysfunctional neuroregulation and a lot of remodeling, such as hyperplasia. And this is particularly all happened because there is an influx of the Th2 cytokines, and particularly the cytokines like IL-4, IL-8, IL-5, and IL-13. The story which I'm going to tell you is all around IL-13. So looking at that, what we thought that, So these are our 3 questions. So our research was being focused on why the people with asthma, they are more susceptible to flu. What exactly happened and why this virus actually exacerbate this asthma? And can we get this into our mice model? So what we did is, everyone used OVA-induced model. This is a baseline model, a universal model being used everywhere around the globe to induce the airway allergic disease, what we call asthma. So what we did is, we actually sensitized the mice with the OVA, albumin, intraperitoneally, and then we challenged the mice intranasally, and then on the day 0, we gave the flu to the mice, which is the influenza virus or PR8. And on the 3rd day post-infection, we cull the mice. When we cull the mice, what we have observed, we observed that the viral titer was really high in the mice, which is having allergy and infection both, as compared to the infection alone. When we looked at the inflammation, it was also been excessively significantly high in the allergic infected mice as compared to allergy and infection alone and control. When we looked at the antiviral cytokines, they all were suppressed, like IFN beta and IFN alpha, and which has been needed to get your immunity. When we looked at the airway hyperresponsiveness in the OVA allergic mice, in the allergic infected mice was too high as compared to the allergy and infection. And the same thing was observed in the hallmark feature of asthma, which is increase in the influx of eosinophils. And they were really very high as compared to the allergy alone in allergic infected mice. When we looked at the levels of IL-13 in the mice, there clearly was very significantly high in the allergic infected mice as compared to infection and allergy alone. And when we looked at the receptor, it was also excessively high. In the histology, so this is a Mason trichome staining, not the histology, sorry. So we clearly been observing that there was a lot of goblet cells and producing a lot of mucus. So you can see that in the allergic infected mice, the mucus production was excessively high as compared to the allergy alone. So this was very clear that there is something happening in the mice model because of the IL-13. So our story started with IL-13. Then we thought to check that, why not we just give the mice pure IL-13 or the recombinant IL-13 and just see whether they are exactly producing the similar features or not. So it was a very short model, and we gave actually, so it was just the very short model. We gave the recombinant IL-13, and on the day 0, we gave the infection, PR8, and on the exactly on the 3-day post-infection, we culled the mice. It has exactly shown the similar results where the viral titer was high in the recombinant IL-13 and infected mice as compared to infection alone. The inflammation was really exactly the same way as it has shown in the ovalbumin model. Both the cytokines were also reduced. Airway hyperresponsiveness was also high. Eosinophils were also high. IL-13 also shown the similar trend, and the psoas with mucus. So the overlaying picture with the OVA model and the recombinant IL-13 alone administration has clearly shown that IL-13 is definitely playing a role here. So what we did is, from our so then we thought, why not we run a clinically relevant model? And the most important clinical relevant model is a house dust mite model, which is exactly resembles the clinics, like a human. So we sensitized the mice, we challenged the mice. On the day 0, we gave the infection, and 3-day post, we culled the mice. It was clearly shown increase in the viral titer. It has also shown increase in airway hyperresponsiveness. It has also shown increase in eosinophils as well as the mucus. And it has also shown reduction in the interferons as well as increase in the level of IL-13, which I have not shown here. Then we thought, why not we just trying to give now anti-IL-13? So that when we know that IL-13 is a crucial factor, which is playing a role here, something increasing the susceptibility to influenza infection in mice, then if we give anti-IL-13, it should reduce all those features. So anti-IL-13 we got up from our collaborators in UK. We used our traditional model of OVA, and on the last day when we gave the infection, the same day we started giving anti-IL-13 till the day 17. And the 3-day post-infection, we call them. What we observed, we can clearly see there was reduction in the viral titer. There was reduction in the airway hyperresponsiveness with the anti-IL-13 treatment. There was reduction in the eosinophils level, and there was reduction in mucus. There was significant increase in the antiviral therapies, antiviral interferons, as well as reduction in the IL-13, which is not shown here actually. Then down the track, we thought, why not we look at the microRNAs level and how this IL-13 actually is related to these microRNAs. When we did our literature search, we have clearly seen that microRNA 21 was one of the factors which was being involved here. And we can clearly see in the recombinant IL-13 and infection mice, the microRNA 21 was really high as compared to the infection and the recombinant IL-13 alone. And when we gave inhibitor, it has suppressed. It has surprisingly, not surprisingly, actually, this is what we were really been hoping that it should suppress. It suppressed actually the mid-21 level. And viral titer was also been highly suppressed. So that was very clear. IL-13 and microRNA 21 are playing an interplay role in this pathogenesis in the cascade of increasing the susceptibility to influenza infection in asthmatic mice. I'm wrapping up this story here because I have to tell you some other story with COPD. So it's still an ongoing study. So what we have clearly seen is with asthmatic patients, there is an increase in an IL-13, and we have also checked this with the facts and clearly shown that IL-13 alpha receptor 1 is a main factor which is involved, and that is actually leading to increase in the microRNA 21 through P10 and increasing the PR3 kinase, which is actually increasing the viral load, and that is actually exacerbating the disease. So this is one side of the study of asthma which is still, we got a lot of data actually, but which is not I have shown, because I thought I'll show you another study of COPD here. So COPD is a chronic obstructive pulmonary disease, and it is basically happened because of smoking. And the smoking is highly prevalent both in Western culture and as well as in Asian culture. Now, it is a chronic airway disease, and the two main hallmark features of this disease include chronic bronchitis and emphysema. This is what it is also known as. If you look at the statistics, almost 64 million people globally are affected by actually this disease. It ranked third in the mortality rate worldwide and fifth in Australia. And it has also led to number of deaths in number of years, and it's consequently increasing. And in India, if you look at the figure, there are almost around 22 million COPD patients, which is costing around 1.1 billion of Australian dollar. What exactly happened in the COPD? So in COPD, it is a disease actually that where actually the airflow limitation normally happen due to the remodeling. This is an alveolar layer and it start breaking. So once it start breaking, the layers break and the difficulty in the breathing will happen because to exchange the air on the surface, they don't have the place. And there's a lot of destruction of parenchyma. So that can further lead to the loss of elastic recoiling and the alveolar region become enlarged. And that has further down the track, it lead to inflammation and mucus as well, which forms a part of remodeling through the hyperplasia. In this particular study, what we have done is we have tried to look at two different microRNAs. One is the microRNA 125A and one is my 125B. So with the previous study and this study, when I will wind up, I'll show you what we're currently doing in terms of drug delivery in my lab as well. So what was the objective? The objective was exactly the same, what I have shown you with asthma. Here also, we were trying to investigate why there is an increased inflammation and the susceptibility to flu in the disease state. The previous one was asthma, here it is COPD. Now, just to summarize, our hypothesis was when a person had a COPD and get flu on top of that, it get increased inflammation. It also get reduction in the antiviral responses to infection, and that can further actually down the track lead to the increased level of these two microRNAs 125A and B. And what we are proposing is it happened through the NF-kappa B and the A20. So A20 and NF-kappa B, they are like what you call, and we call in inversely proportional. So if A20s go up, NF-kappa B go down. And when NF-kappa B is up, A20 is down. And remember, NF-kappa B is a co-mediator. If it always goes up, inflammation goes up. And that can lead to the inflammation. The other factor what we look on our study is called mitochondrial antiviral signaling, called MAVs. And that MAVs also lead to the inflammation. So what we are proposing here is when we inhibit the influx and the increase of the microRNA 125A and B, it should reverse the whole cycle. And once it will reverse the whole cycle, it will reduce the inflammation and actually it will improve the pathogenesis or the susceptibility of influenza infection in COPD. So let's see what the data we got here. So this study is carried out both in human cells as well as cells which are obtained from humans, as well as in the animal models. So we can clearly see here that in the 6th hour and the 24 hours, so you can see there are 3 groups here. One is the healthy, which is a normal control. The other one is a COPD, which is a person having a disease, but the person don't smoke. And the third one is a smoker, which only smokes, but doesn't have a disease COPD. So it is actually control for, smoker is just a control for COPD. So when we have looked at these cells, it has clearly shown that the inflammation was really high. So in COPD, you can see the inflammation both at 6 hours and 24 hours was really high in terms of P65. And when we compare the IFN beta, which is the one of the antiviral, interferon, it was reduced in COPD through the Western blot. And this is in the human cells. And that has been obtained through the bronchoscopy in a John Hunter Hospital by Professor Peter Walker, who is a clinician. Then what we did is we also looked at A20, because we are hoping that it is all being the NF-kappa B is just being mediated through A20. So A20 was actually being reduced in COPD. So you can see that the middle one, the bar graphs are less than as compared to the healthy and the smoker. They, with the Western blot, both had the 6 hour and 24 hours. And these were the time points were decided on the basis of our previous studies. And to confirm that, we have done the immunohistochemistry, and it was very, very obvious. So the more the brown color on the epithelial cells, the more the A20 is present in the airway. So this is control. That's the COPD mice. This is with the infection alone, and this is a COPD plus infection. So you can see that the brown color appearance on the epithelial cells was significantly high in the control as compared to the COPD alone, infection alone, and COPD and infection. So it means the A20 was reducing with the disease and disease plus infection. And that was very much consistent with the results what we got with the human cells. So what we did, the other thing we looked is what happened to these microRNA we talk about, and we are thinking with 125A and B. These two microRNA in the human PBACs, or what we call primary bronchoepithelial cells, they were increased. They were really very high as compared to healthy and smoker groups. And that was very obvious. These two microRNAs are really playing some role here. And this influenza infection actually had led to a significant decrease in these levels when we treated with the inhibitors. These inhibitors are also called as an antagomere. So when we gave the inhibitors, the level of these two microRNA was suppressed in the cells. So it means the inhibitors were really good. And let's see how it worked further down the track. So when the inhibition was happening of the 125A and B, so what, according to the hypothesis, the A20 should go up. And this is what exactly happened. The A20 level was really up. And that has also significantly enhanced the interferon level, IFN beta. So that was a double thumbs up for us. The story was going in a right track. Then down the track, we thought, why not we check this in our mice model? So this is a very unique mice model, which only 2 labs all over the globe is running. And one of my own mentor with whom I did my second doctoral studies, and because my study also form a part, and this is some of the data is coming out from that as well. So what we do here is, this is called a nose direct smoke or nose only smoke, because to induce the COPD in mice, people around the globe, what they do is they put the mice with the whole mice into a chamber and they put the smoke in that, which is called as a whole body exposure, where the mice doesn't have opportunity to directly breathe through the nose and hold smoke, which goes to the lungs and create the disease. But in this model, it was a nose only smoke induced model, where the mice actually, so it's a whole lot of a chamber, and the mice being normally been put into a tube, and they've been put on a tower, and in the center of the tower, the smoke comes, and what it mice inhales. So the mice were being smoked for 8 weeks, and on the last day of the infection, we have on the last day of the smoking, we have given the infection, and on the 7-day post-infection, the mice was culled. And when we cull the mice, what we can clearly see in the smoke mice, the level of the inhibitors of 125A and B, they were significantly high as compared to control. And when we looked at their body weights also, so during the passage of the smoke, they were reducing. And when it started giving the inhibitors, their weights started getting increased. So that was very obvious that microRNA 125A and B and antagomeres have decreased the weight loss as compared to the scramble treatments, or scrambles are just the controls. When we looked at the histopathology of that, so this is a very typical histopathology of lungs. You can see various airways and a parenchyma, and you will see some blood vessels as well. These are the blood vessels. So this is control, or so this is scramble is a control, this is a air group, and this is a smoke with different treatments. So you will clearly see that with the scramble in a smoke, there was an excessive inflammation as compared to the air or the control mice. Down the track, actually, when we have treated with the microRNA 125A and B inhibitors, it actually had reduced the inflammation. And when we use the combination of both the 125A and B, it has further enhanced the inflammation. So that was very obvious that 125A and B is doing something which is helping in reducing the inflammation. So what we did after that is we thought we have to look now on the levels of the A20. And as I showed you the immunohistochemistry previously, where the increase in the brown color or intense color around the airways in the epithelial cells clearly defines the existence of A20. So when you clearly see that with the scramble, there was very less brown color, but when with the treatment of the antagonistes or inhibitors, the brown deposition of the color was really increasing, and that was clearly showing the extensive presence of A20. And when A20 increases, it automatically reduces the NF-kappa B. And we have also checked through the Western blot, the A20 levels were really up, and the antiviral interferons were also up. So that was very obvious. When we looked at the cytokines in the same model, we can clearly see it has increased IL-6, Kc, which is also known as CXCL8, which is well known in COPD, TNF-alpha and IL-1 beta, they all were up in the disease. And on the treatment, they were suppressed. And the same thing, when we looked at the viral titer, it was also been inhibited or been reduced when being treated with the antagonistes as compared to the scramble or control. Now, after this, what we thought is the same thing we need to actually look in the mice in terms of the MABs. That is the another pathway we thought of. So, So what we thought is because the mitochondrial antiviral signaling also actually has led to the increased induction. And that also had led to the increased level of the interferons as well. And that has also reduced the viral replication. So because all these things were happening very clearly and obvious, and that was very clearly defined to us that MAVs, and which is the mitochondrial antiviral signaling, along with the microRNA 125A and B, are really playing this role around the whole cycle. Now, in terms of concluding this study, so what we have done is we know that in both the patient cells, which is obtained from patient, the primary bronchoepithelial cells, as well as in the experimental model of COPD, when we give the flu on top of that, that has definitely increased the level of microRNA 125A and B, which are the important regulators of the inflammation. And that has been controlled by two factors through our study, A20 and MABs. And they have literally reduced the level of A20 and MABs. And that also, once they have reduced these factors, it has actually led it through the increase in the NF-kappa B. When it has led to the increase in NF-kappa B, it has increased the inflammatory cytokines, what we have seen like IL-6, IL-8, CXCL8, and all those. And what we have seen as an outcome of this study is once we inhibit the increased influx of these two microRNAs, 125A and B, it actually reversed the whole cycle. It actually reduced the induction of the inflammatory cytokines and improves the antiviral responses in an infection, IAV infection in COPD. And that clearly has shown that this identifies a potential therapeutic target in flu or influenza infection-induced exacerbation of COPD. This study has already been published in a very high impact Journal of Clinical Immunology Inside, JCI Inside, and it's already well known. So as in my lab or the group which I have, so we work on both the drug delivery and the biology side. So currently, one of my PhD students is actually preparing nanoparticles, have prepared already those nanoparticles with the encapsulating these inhibitors of 125A and B, and also certain oligonucleotides. And we have clearly tested these on the basic cell lines of the lungs, like BES2B, which is the bronchial airway epithelial cell line, and also on A549. And they have clearly shown that they target majorly two cytokines, IL-6 and IL-8, which are actually down the pathway, actually control the inflammation. So this study is still actually in terms of the drug delivery, we are still pursuing, but in the terms of the cellular and molecular pathway, it is still, the asthmatic one is still an unpublished, but the COPD one is already published. Apart from that, my group also works on certain phytoceuticals and the medicinal plants of importance, what we say. Those we normally encapsulate, and we also look on these different states in lungs, including infection. Now, when a common question which I normally get in most of the conferences is, can this be also this cycle also forms a part of COVID? 100%. So the answer is yes. These are the core microRNAs, not the 125A and B, but 21, yes. You will see 21 is actually well known in the various cancerous states. But in terms of the 125A and B, it depends, because it varies from disease to disease states. This is what we have looked in respiratory, but you will see microRNA 125A and B are also being in colorectal cancer. So it depends. And always remember, when we talk about antagomeres, they always have off-targeted effects, which we don't want. So there are a lot of still hassle going around when we are trying to encapsulate that into nanoparticles, and the base nanoparticle we used are the liquid crystalline. Sorry, excuse me, not the liquid crystalline. Liquid crystalline we use more for the phytoceuticals. It's the sugar-based nanoparticles, or what we call dextran-based, or the polysaccharide. And the reason we do in those is because macrophages love sugar-based nanoparticles. And macrophages, as in the previous talk by Dr. Dubey also, it was very obvious that they are one of the core cells in inflammation and in respiratory diseases, and always good to target. So that can also, be done through those various kind of the novel techniques in the drug delivery, what kind of the nanoparticles we are being using, and how we are preparing, and those kind of things. So in terms of the phytoceuticals research, we normally use liquid crystalline nanoparticles or lipid-based nanoparticles to encapsulate them. We have worked on various drugs such as rutin, berberin. Everyone worked with curcumin. We also started with curcumin, naragenine, celestrol. We have worked with boswellic acid. We have worked, now currently we are working with two new drugs, Zirumbun, we got from our collaborators in Japan, and also we're working with agarwood, because I'm working with a company here where we have developed cosmetics-based products with agarwood oil, particularly hand sanitizers and some face creams. And now we are testing the antiviral and antibacterial potential in terms of, and also how it can be modulated if it needed in lung diseases. So this is a core area I work, and I have shown you a bit of my research in terms of the COPD and asthma, what we do, and what the models we have in our laboratory. And these are in collaboration all with my mentors, as well as which currently I'm doing as well. So with this, actually, I would like to acknowledge some of the important people, because this was a funding from NHMRC and this project. And I would like to thank Professor Phil Hansborough, Alan Su, who have done most of the studies, help with the human work, and Malcolm Starkey. I would also like to thank particularly the school where I'm currently based at. The Graduate School of Health, Discipline of Pharmacy. Charlie was the founder dean of the school, and then Kylie is currently the head of the Discipline of Pharmacy, and Mary is one of my collaborators. I would also like to thank actually the organizing committee of ISTE AICTE sponsor this program for the online refresher program, and the College of Pharmacy, Tatya Sahib Kore College of Pharmacy, particularly Professor D'Souza, who has invited me to talk about my research on this platform. It's really an immense pleasure, and thank you so much, Professor D'Souza. I've been already working very closely with Professor Patravel, who is here, and it's an honor to see Professor Patravel here as well. And Puppet is there. I've been working with him and Professor D'Souza, and also I know a few people I've been working with closely here. So it's really an immense pleasure seeing you here, and really looking forward to continue that and seeing how we can convert this translational research actually to commercialization so that it can reach to patient. Because no point working on the research just for the core of sake of the lab work and getting thesis done or paper done. It is really important we should contribute to the community in one or the other form, whether it can get into the industry or commercialization, or in any way, if it is giving a social impact. That is what my core lab always had a principles. Even if it is helping a student growing the career, and if it's sticking that way in this project, that makes me feel comfortable, and that makes me feel quite happy about it. And with this, actually, I would like to thank once again each and everyone, those who have attended my talk very patiently. um, it says in Australia it's summers, but it doesn't look like summer. You might be seeing me sitting in a jumper at the moment, but normally at this point of time in Australia, we have bushfires going on, and there was really bad last year. While this year, I believe the climate change has played very well. And now we have, the summers are not coming, and every day just looks like a normal average winter day. So with this, thank you so much. And if you are anytime here in Australia, please give me a buzz. I'm more than happy to host. Thank you.
Thank you so much, sir. I request delegates, if you have any question, queries regarding the topic sir have discussed, then kindly unmute yourself.
Hello.
Yes, yes. Hi, Dr. Solanki.
Yes, yes. Very excellent presentation, sir.
Thank you. Thank you.
In this, I have one query. Like,
Yep.
how will we establish the correlation with the different types of mRNAs and with the cytokine levels that you have measured in the viral infection model of the mice?
Yeah, so
Is
it
there
is,
any specific method?
sorry.
Any specific methodology that the correlation and the establishment can be possible?
So, indeed. It's a very good question, actually. Thank you, Dr. Solanki. So what it happened is we really need to go to the genomic testing kind of a thing for these levels, because what we have done is a baseline things, correlating microRNA to the cell, to the inflammatory, cytokines or chemokines or whatnot. But to do that, we definitely need to go into the, what we call is the bioinformatics. Particularly that kind of the tool will actually help you, and we have done that. Means I have not done in the group. Actually, there was another student who have another person in the group who had done actually correlating what kind of the microRNAs are actually targeting which kind of the cytokines. So the bioinformatic analysis and tools are actually the one which actually help to first to check out theoretically, and then to check on the same models. So what we do normally is we check on both the, what we call is bronchoalveolar lavage fluid, BALF, as well as on the lungs, homogenates in the protein. So it need to be done on both. So if it really shows the increased influx of those cytokines in both, then it's always, it says a double thumbs up kind of a story. But if it doesn't shows a correlation, which normally happen, so then we need to definitely go back to the literature. But 100%, and most of the time what it happen is, you will see these microRNAs are more known in cancer than any other disease states. You will see any microRNA you pick, they would be always been well established in one or the other cancer. But when you look specifically on any diseases, if we talk about asthma and COPD, you will see microRNAs, but they would not be as significantly or predominantly said as it's been said in cancer. So I hope that makes sense, Dr. Solanki.
Yes, sir. Thank you.